I'm going to talk to you for 3 minutes about rogue proteins. The one that we hear the most about is a protein called a prion, and that's why I'm here and why the Institute was created. Prions are proteins that occur normally, which misfold and, as I said, go rogue. And they become infectious in that rogue form and cause enormously debilitating and deadly neurological diseases. They're really unusual, very unusual infectious agents. In 2003, when one of the prion caused a cow, a dairy cow in northern Alberta, to have bovine spongiform encephalopathy, but in the middle of the press conference where Canada was announcing this, borders were closing to a $4 billion a year industry. So hence the creation of the Alberta Prion Research Institute to have in Alberta an ability to be able to respond scientifically to these kinds of challenges, because no such ability existed before. I want to tell you this morning, however, about a new connection that Alberta might make, since this is the Connect conference. 50 years ago, Alberta began to build and continues to build a world-class presence in protein structural research. 35 years ago, Alberta began to build and continues to build a world-class presence in neurosciences. 5 years ago, Alberta began to build and has actually been relatively successful in building a world-class capacity to do prion and protein misfolding research. The folks who actually generated the paper that created the Alberta Prion Research Institute were actually quite prescient, because they looked and they said, well, yeah, prions misfold and they cause these plaques in brains. But other neurological diseases cause plaques in brains, and they're probably occurring because proteins have gone bad. So we were given the mandate to study prion and other protein misfolding diseases, which we have done. Now, the prescience was greater than that. I think they sort of figured, well, maybe there's a connection. The last 3 years, science has now taken us to the point where we now know that in many of these other diseases, the proteins are probably misfolding in the same way as prions are. A simple example, this is a normal protein and it goes bad. This is another normal protein and it goes bad. And then they get together and they form aggregates. That's what everybody thought about most of these diseases. For prion diseases, we know the normal protein goes bad and it meets another normal protein and causes it to go bad. Now, that may not mean a lot to those of you who are not mechanistically inclined, but if you are, it's an enormous difference and it presents all sorts of new ways of thinking about how you deal with the disease. Recent evidence has shown us that Alzheimer's disease, Parkinson's disease, Huntington's disease, frontotemporal dementia, and probably other diseases may well be following the prion mechanism. So, Alberta is now positioned, having built 3 great streams of research to connect them, to merge them into a very strong river of research. We often talk about serendipity and the prepared mind in science. So the question is, we've now had a serendipitous set of occurrences happen to us because we are now positioned to be among some of the world's best equipped to study not only prion diseases, but now the mechanisms of some of these other dementias. So the real question is, will the system be prepared to take it up? And more importantly, will those of you young folks who are here be willing to think about it, and will you be prepared to take it up? Thank you.